Hello everyone, I hope you're having a great day. So in today's video, we're going to be going into and beginning the muscular system. So this here, we're kind of focused on the skeletal system here for how a skeletal muscle contracts and all the major skeletal muscles of the body. So this first little video, we're going into an intro to the muscular system where we're covering muscle tissue anatomy. Again, the focus is on skeletal muscle anatomy here. So in this video, we're going to describe the different types of muscle tissue. So we will give a reminder of that histology chapter of skeletal, cardiac, and smooth. And then we're going to look at skeletal muscle specifically, look at their layers, the connective tissues, and then go down into the microscopic anatomy of that skeletal muscle. So I know this is a survey course, but one part of this is that I want you to understand how a skeletal muscle contracts, and we need to understand this anatomy of the skeletal muscle in order to understand skeletal muscle contraction and the sliding filament model for muscle contractions. We got to understand the proteins and that microscopic environment inside those muscles. All right, so let's get right into this here. This first part is just a little refresher from histology in the three different types of muscle tissue we have. So right here we have skeletal muscle. The first one here is skeletal, and we remember this because you see the striations, the striping patterns. We'll go over details on what causes those striations again, and you see they are multinucleate and very long. And then here is smooth muscle. So here we have an example of smooth muscle. You can see the nuclei right here, and they are sheets of cells that line up. And within smooth muscle, there are no striations. Now, they do have actin and myosin filaments. However, they overlay in a slightly different manner, and they have a different ratio, so they do not have striations. And then down here is a representation of cardiac muscle. And remember, smooth and cardiac are both involuntary. Smooth is the in all hollow organs except the heart, and then cardiac is found in the heart. The heart is cardiac muscle is uninucleate, and then also it has intercalated discs, which are the gap junctions, and not sure one is here, but maybe there's one there, and then that would be the gap junctions between cardiac cells. It looks like there might be one there, hard to tell in this particular histology images. There are some better examples out there than this one, but this little first part here was just a refresher. There are three types of muscle: skeletal, cardiac, and smooth, and we are focused on the voluntary skeletal muscle contraction and the anatomy of those today. So here, looking at the connective tissue coverings of skeletal muscle. So here, gross anatomy of a skeletal muscle. As we work our way deeper and deeper and deeper, the outer layer of the muscle has a connective tissue on it, and it's called the epimysium. So whenever you see MYS, it means muscle, mysium, or epimysium here. And then the way the large muscle arranges itself is that it creates these little pockets, these little sections or these fascicles. So when you break down a large muscle, you kind of see like string-like patterns in it. I'm sure you've shredded pork or chicken before, and now those strings you see are the muscle fascicles. And then surrounding each of those fascicles is a connective tissue as well called the perimysium. So peri is around, epi is upon, peri is around. And then within the fascicle itself, this is now where we have the individual muscle cells, or what we refer to as muscle fibers. So a fiber is equal to a cell in this regard. So if we look at an individual muscle cell, then we see all these little rod-like elements inside here. So imagine these rods all running the length of this. And so those are called the myofilaments. There's an actin and myosin filaments, and then make up those myofibrils. Inside that then are the myofilaments, which are the actin and myosin. The rod itself is the myofibril. Sorry, got that terminology backwards there. And then there are also supporting cells around them called satellite cells as well. So here's when we think of cells, you usually think of a nice little nucleus in the middle and all the organelles. Muscle cells or muscle fibers are very different. They're just packed full of these myofibril rods. And they're also surrounded by connective tissue out here. Well, this one doesn't show it. This one shows a cell membrane called the sarcolemma. This is another root word for muscles, sarco. Whenever you see sarco, it's talking about muscles. And then the connective tissue layering on that one is called the endomysium. So there's a connective tissue layering around every individual muscle cell, and then every muscle cell, of course, has its own cell membrane called the sarcolemma. There's a structure inside muscle cells called the sarcoplasmic reticulum. There's the sarcoplasm, that's the cytoplasm. The sarcoplasmic reticulum is the smooth ER, and it's an important part for calcium storage. So some of the terminology does change from that stereotypical cell we're used to talking about. All right, so let's look at the anatomy inside one of these myofibrils, and we talked about those myofilaments that are found inside. So if we look deeper into that muscle fiber, here again is that same image where we have that myofibril sticking out. We have the sarcolemma, a lot of mitochondria in there. Mitochondrion is just singular form of that, a lot of nuclei, and so forth. We're going to talk about these bands that appear, but these are what the striations are. You see this darker band. It's called the A band. There's an A in dark, and here would be a light band. There's an I in light. So there's these alternating bands that lead to the striations in the muscle cell. And so what causes those banding patterns? Well, it's the overlapping thick and thin filaments. So there are two types of filaments here. There's a thick filament made of myosin, and then there's a thin filament made of actin. And so just to kind of represent this here, all the thick filaments in this image are shown in this purple color, but I'm going to color it red right here. So just to color a few in, just to make it a little easier to see, we have these thicker rods right there. And then we bring in maybe a light blue here. Let's see, is this thin enough? Yep, let's do that. And these blue filaments here are then actin, and they kind of run right through here as well. And so they are thinner. That's why they're called thick and thin, but they're made of actin, and they are held together by this disc in the middle here. It's referred to as the Z disc. So Z disc to Z disc is defined as one sarcomere. So here's the definition of a sarcomere. There's that sarco term again. So Z disc to Z disc is one sarcomere. That's one train car. And then think about a whole bunch of these train cars. So there would be Z disc to Z disc in that image connected together. I have an image of a muscle cell down here from a laboratory model. And so right here would be one Z disc, and right here would be another. So you can see all these train cars sort of lined up with each other. So just get an idea of how microscopic we are looking here in one of these myofilaments in these individual proteins that make them up. And as you can see from all the dots at the end here, there are a lot of proteins. You got to remember this is also a 3D structure we're looking at. Now back to some of the labeling here. We have the dark band. This band right here is considered dark, or the A band like we mentioned, because it is all myosin. So if you take a section right here, it would be all myosin, and all myosin, making it appear darker. Whereas over here, this section in here is only actin. There's no myosin found there, so that a band appears lighter, or it's known as the I band. There are some other band patterns here, like right here is the H zone. The H zone is where it is myosin only. There's also the M line, which goes down the middle right here, and there's a structural protein in there. It's labeled over there, called myomycin, which helps structurally hold that together. There are some other proteins found in here as well, such as titin, dystrophin. Muscular dystrophy is a disorder affecting that dystrophin protein. So there are other proteins involved for the structural integrity, but primarily muscle proteins are made up of these actin and myosin filaments. So pretty wild how it is. Just so when muscle contracts, the muscle shortens. So here this slides in, and this part slides in. We'll talk about how that happens and how that occurs coming up. Oh, and one way I remember H zone, heavy. This is all heavy bands, or all thick bands right there. Apologies for the barking dog. Mail just arrived. Good timing. And then here's skeletal muscle. And I like to show one muscle fiber. Every single muscle fiber has a motor nerve going to it. And so this is the motor neuron. We didn't go over the structure of these yet, so it's okay, but this is how we activate the muscle. So every movement I'm doing in my hand right now, doing these drawings, this movement here, I'm activating skeletal muscles to do that, and I'm sending this signal at this neuromuscular junction to transmit that signal from my nervous system saying to move to activate that muscle and then cause a shortening of that muscle. So every single sarcomere would then shorten, so this entire muscle cell would then shorten itself. So every individual muscle fiber is activated by independent motor neurons. So when you do a biceps curl, you might activate a certain amount, and when I pick up this pen here, I don't activate as many. So you can individually control all of them. And so one big important part in this chapter here is going into the mechanism on how we send this signal. It's called excitation, contraction, coupling, and then the mechanism within this muscle cell for how contraction works. And that's why this video is included in this unit, so that we can understand that link to the anatomy and contraction. And then one last little bit here, little zoomed in version of those proteins, the thick and the thin filaments. Here's the same lines that are labeled. And the big thing is that on those myosins have all these little heads sticking off of them, and it's 3D. These heads have 2 binding sites on them, and they bind or want to bind to these binding sites on actin. So the thick filaments have these heads that come up and bind to actin, and then they go through something called a power stroke, and that's what leads to shortening. And we're going to go into detail of this a little bit more in the contraction chapter. But here, like in this image, all of these myosins would have heads that can come up and contract actin. I like to use my arm as an example here, where the myosin head binds to actin, goes through a power stroke, binds again, goes through a power stroke. And so all that's happening right here, and that's what leads to a shortened muscle. So really, really cool when we get into that, and that'll be coming up later. Another thing I want to mention with muscles is, how do you send a wave of action potential and excitation to the middle? So yeah, if you send an excitation up here, you might contract them, send calcium to these fibers or these filaments here, and boom. But how do we contract deep in? How do we send this wave all the way around and also go inside? Well, there are these structures called T-tubules, or standing for transverse tubule. So the sarcolemma actually has these little pores. The sarcolemma is the cell membrane that then travel down inside and around the individual contracting units. And then around each T transverse tubule here, there are what are called the terminal cisternae. These are parts of the sarcoplasmic reticulum which hold calcium. I know it gets a little complex right here, but in short, the excitation comes down. It causes a calcium release from the sarcoplasmic reticulum, and the calcium is then what leads to a muscle contraction. And so without these terminal cisternae in these T-tubules that form this little triad substance, you would not be able to send action potentials deep into the muscle cell and release calcium throughout the entire muscle fiber. So here you want to go all the way down in and be able to activate the internal muscle fibers, or myofibrils, rather than just the external ones if the wave traveled around the outside. So these transverse tubules come in and wrap around all the individual myofibrils so that the myofilaments can contract, which are the individual rods inside there. So I know that was a lot to start this chapter, but it's really important that we understand this anatomy before we go in to the actual muscle contraction, and then we'll go over all the major muscles of the body and their functions as well. All right, that's all I have for today. Hope you have a great rest of your day, and always let me know if you have any questions. Bye-bye.